Hello and welcome to our lecture on Introduction to Mechanobiology. So, over the last 2-3 lectures we have been discussing about actin dynamics in migration. Okay. So, I have introduced you to the technique of fluorescent speckle microscopy for tracking dynamics. Okay. And in this regard, we were discussing a paper where fluorescent speckle microscopy was performed to track or quantify actin monomers movement during migration. Okay. So, based on that, the authors for cells using epithelial cells. Okay. So, authors showed the existence of two zones, lamellipodia and lamella. Okay. So, in lamellipodia you have speckles, fast moving, short living speckles, while in lamella you had slow moving, okay, long living speckles. This was one difference. So, lamellipodia is within first 1 to 2 microns from the leading edge. So, if this is a cell, you have the lamellipodia as this zone in the front. Okay. So, this is your lamellipodial region and then this is your lamellum. Okay. This is your lamellipodia and this is the migration direction. Okay. So, speckles, all these speckles move backward. So, this is termed retrograde flow, retrograde or backward flow. Okay. What I also told you was that these two networks, lamellipodia and lamella, are molecularly distinct. Okay. So, and using blebistatin experiments, the authors had shown that myosin II in the lamella contributes to retrograde flow. Okay. So, if you treat cells with blebistatin, which is a myosin II inhibitor, then the retrograde flow in the lamellar region is eliminated, but the retrograde flow in the lamellipodial region still exists. Okay. So, the last thing we discussed yesterday was by taking a cell edge, okay, we wanted to know, okay, how actin dynamics is contributing to edge displacement. Okay. So, what the authors would do is they would discretize this network. Okay. So, you would discretize this network into small zones. Okay. So, you have these zones labeled from 1 at one end to whatever, let us say 40 at the other end, okay, and you would plot. So, your x axis is time, okay, and y axis is whatever you are plotting, but along the edge. Okay. So, if locally, let us say, so this position is 1 and this position is 40. Okay. Let us assume at position 10 you have a polymerization event. Okay. At position 10 you have a polymerization event. So, if you are plotting the network turnover, so if you are plotting turnover, okay, so let us assume at position 10, position 15 and 5. Okay. In position 5, 10 and 15 you have polymerization events. So, what you will have at this position 5 at this particular time t, you would have a green signal here indicative of polymerization, at position 10 also a green signal and position 15 green signal, and at all other points you will have red signal. Okay. So, this allows you to capture what is happening at this particular time instant along the edge. So, this may vary in the, so if this is the dynamics that is happening on the along the edge at this time, the next instant you might see that in the neighboring point you might see a green here and red here. Okay. So, suggesting that in the next point, next time instant there is a depolymerization event, so on and so forth. Okay. Or you can have prolonged polymerization. Okay. You can have multiple time points along at this particular point, let us say at this point you see polymerization happening continuous. Okay. So, this is turnover. You can generate the same plot for let us say edge displacement. So, if the cell is migrating this way, then this direction is positive and this direction is negative. So, this direction is negative, this direction is positive. Okay. So, you can have a length scale where red is positive max, blue is negative max. So, you have a continuous scale, and just like you did this, you can do the same thing. So, at this point let us say the cell is migrating fast. Okay. So, if I were to go with this thing, then at position 15 okay, I would see max. Why? There is a polymerization event, I would see high displacement, high edge displacement in the positive direction, and where there has been depolymerization I would see negative, which means that the cell is retracting. Okay. So, these suggest that these three things are all correlated. Okay. These three things are all correlated. Okay. So, you can target, you can track the coupling between these individual components by doing cross correlation. Okay. So, you can do cross correlation to study the coupling between edge displacement network turnover and flow. So, let me draw a sample plot as to how this cross correlation might look like. Okay. So, you have time here, this is your cross correlation. Now, the way cross correlation is defined, okay, so let us say this is my 0 and this is my 0, this is 0, or you can say lag time, okay, and this is 0, correlation, cross correlation. So, you can have cross correlation of positive or cross correlation as negative. Cross correlation as positive would mean between any two quantities would mean that one guy, if it is positive, the other unit also would be positive. So, what the authors found, okay, so they did cross correlation between three things. Okay. So, let us say, let me call the edge displacement as E, okay, the network turnover as T and the flow as F. Okay. So, what they found, okay, let us first plot E versus T, and what they found was this curve looked something like this. Okay. So, if you notice the way I have drawn the curve, so suggests that there is at one point there is a positive correlation, at other points is mostly 0 or baseline. Okay. Now, let me draw the curve E versus turnover. Okay. So, what you find, the E versus turnover gives you a peak here. Okay. So, this is E versus turnover, okay, and this is E versus, this is F versus, sorry, this is not E versus turnover, this is F versus turnover. So, F is flow. Okay. So, F and turnover are negatively correlated, while E and turnover are positively correlated. What you also see that the peak, the positive peak between E and turnover, when they are related, is slightly to the left. So, peak of E versus T is slightly to the left of F versus T, suggesting that if I were to imagine the sequence of events that are happening temporally, there is a polymerization event, so which would mean as turnover. Okay. That polymerization event is first pushing the membrane, or in other words edge displacement is positive. So, you have this positive correlation in the red curve. Immediately after that, there is a negative correlation between F versus T. Okay. So, so turnover is positive, so you have a polymerization event, and flow is negative, which means that there is retrograde flow. So, what is happening is that polymerization pushes the membrane, okay. This is my first step, okay. And right after this, okay. So, if I were to think of Newton's laws of motion, to every action there is an equal and opposite reaction. So, if the actin filament, the polymerizing actin filament pushes the membrane will push the filament back. So, membrane pushes the filament in opposite direction. Okay. So, as a consequence, and what will happen if the membrane pushes the filament back, then this should lead to retrograde flow. Okay. So, this is, so once again this is the time sequence of events in terms of cross correlation. You have a peak in edge versus turnover, and right after that positive peak, when the edge moved ahead, then the edge, so your edge moved ahead and again the edge, your correlation dropped. There was a corresponding flow, and this flow was backward, because the turnover is positive, the flow is backward, so you have a negative correlation. Now, what happens to this curve if, okay, if cells are treated or perfused with Cyto-D, with the drug Cyto-D, right, cytokinase D. Okay. And what does cytokinase do? Cytokinase treatment actually inhibits the polymerization of filaments. Okay. It prevents Cyto-D, this guy prevents actin polymerization. Okay. Okay, so in terms of cross correlation, okay, you have the following. Okay, this is lag time and this is cross correlation. So, this is 0, 0, this is cross correlation. Okay. So, what you have, okay, your red, which is the edge displacement, okay, there is no correlation between E versus T. Okay. But if you do the correlation between edge displacement versus flow, okay, what you will observe is a curve which is like this. Okay. In other words, edge, so your flow network turnover is mostly negative because there is no new polymerization. Okay. This is negative, okay, and so we are plotting E versus F, okay, E versus flow. Edge displacement is backward. Okay. So, if I were to draw the curve, what is happening here is after cytokinesin D, okay, if this is your leading edge, this leading edge at different time points, it is retracting, it is coming. So, this responds the leading edge. Okay. So, leading edge is retracting and it is correlating with the retrograde flow, is correlated with retrograde flow. Okay. So, what you have, this suggests that cytokinesin D eliminates, okay, eliminates the lamellipodia. Okay. So, then in that case, what drives the retraction? Retraction is driven by the lamella. You have myosin 2 in the lamella, which is pulling the filaments back. So, earlier you had two components. At the leading edge, that pushing back was provided by the membrane. When you eliminate the lamellipodia using cyto D, what you are left with is retraction of the cell edge that is driven by the lamella. Okay. So, this would suggest that if I were to once again draw the leading edge and let us say this is my interface. Okay. So, if I have these discretized zones at the beginning in the lamellipodia and then I extend this to these discretized zones right behind the lamella, okay, right behind the lamellipodia along in the lamellar region. Okay. So, if I see that how is velocity at this point correlated with velocity at this point. Okay. At each point I can see the flow in the leading edge and see how is it correlated with the flow in the lamella. So, what we observe, okay, so once again this is time, okay, let us say every time the velocity is, so because flow is backward, your velocity, this scale is actually negative, okay, it starts from 0, let us say this is minus 15 or whatever, okay, you have velocity. Okay. So, every time you have the flow at the leading edge, okay, what you see is, sorry I drew this wrong, okay, the flow in the lamellipodia okay. So, the way I have drawn it is the flow at the leading edge seems to be exactly in sync with the flow at the lamella. So, there is a periodicity of edge movement and retrograde flow. So, this points out to a periodicity of edge movement and retrograde flow. In other words, whenever the edge moves back, the lamella also moves back. Okay. So, this suggests, so there are two possibilities, okay, that is it that if these were perfectly correlated, is it that the protrusion, so you have the leading edge, okay, you have the leading edge, let us say at a given time step, from this you had this local protrusion. Okay. So, I am overlapping the membrane, leading edge membrane at two different configurations. This is T plus delta T and this is T, this is at T, the cell is moving in this direction. Okay. So, for this protrusion to happen locally, one possibility, okay, one possibility is the lamellipodia expands. Okay. One possibility is the lamellipodia is expanding, or the other possibility is the lamella is expanding. So, what is the difference between these two possibilities? If the lamellipodia is expanding and if this protrusion is very local, this would mean that the lamellipodial thickness will increase, versus if this was true that the lamella is expanding, then whenever you have a protrusion event, independent of the protrusion event, the thickness of the lamellipodia will remain the same. Okay. So, what the authors found was the lamellipodial thickness remains unchanged. Okay. So, this would mean that the lamellipodia is expanding and any local deformation, okay, is associated. So, edge displacement followed by expansion of the lamella. So, how is it working? Okay. So, in other words, okay, so what you see, if this is the leading edge at one time point, okay, this is you have the, let us say you have a local increase, okay, you have a protrusion in this direction and this is associated. Okay. So, I am drawing the initial position. So, this was the initial position of the lamellipodia, okay, and it extended to here. Okay. So, it is not that the lamella, this position of the lamellipodia remains the same, rather the position of the lamellum increases. Okay. So, now in the case of protrusion, this whole thing will represent the lamellum. So, in other words, at this site you have a lamellar expansion. So, how come at this you have this continuous retrograde flow, you have something flowing ahead. So, actin filaments polymerizing, pushing the leading edge forward, the membrane pushing it back and it is retracting. So, if that would happen, then the overall position of the cell will not change. It is like a curtain which is blowing in the air. Every time the wind hits it, the curtain moves. Once the wind is gone, the curtain goes back to its original position. But every once in a while you have this lamellar expansion which leads to stabilization. Okay. What is happening? Okay. What is the difference between leading edge movement happening at some points and at other positions you have this fluctuation of the leading edge? Okay. So, what the authors found? Okay. So, I have drawn 3 points, let us say, where the leading edge extended. So, what the authors found? So, let us say this is the position of the interface. Okay. So, what the authors found were wherever this protrusion was stabilized, the authors found that focal adhesions were formed right behind the protrusion in the lamellar region. Okay. So, this suggests that formation of focal adhesions drives leading edge movement. Okay. So, to summarize, so these observations, okay, so based on this we can summarize the data as if this was a cell membrane. You have 2 networks of actin filaments. Okay. One is here, the other network is here. Okay. And then, okay, so there is some overlap, and at these positions of overlap you have the formation of focal adhesions. Okay. So, this is your lamellipodial network, lamellipodial actin network. This is your lamellar actin network, and this is your focal adhesion. Okay. So, suggesting that in cells migration is driven by the existence of two kinetically and kinematically distinct actin networks. So, these observations also led to the to our understanding of migration, saying that focal adhesions act as molecular clutch. Okay. So, if I were to draw the membrane of the cell, okay, you have these integrins, let us say these are my focal adhesions. So, if you have an actin filament, okay, this is an actin filament, and you have monomers which get added to the front edge of the cell, right. So, in the absence of this connection, okay, so in this case when these actin monomers get engaged, so at the rear end, okay, so in this filament you have myosin which pulls on this, okay. So, causing this myosin pulling on it causes retrograde flow. So, if the actin filament is not attached to the integrin or in the disengaged configuration, you will not have any edge displacement, but you will only have retrograde flow. However, if the focal adhesions are engaged, okay, so if the focal adhesions are engaged, okay, if the focal adhesions are engaged to the actin, okay, so this is my actin network now, okay, you can have filament polymerization at the leading edge, okay. So, this is your protrusion, and the myosin motors pulling on the filaments will cause traction at the base. Okay. So, this is called a molecular clutch, okay, molecular clutch. It is similar to our cars, that if your clutch is not engaged, then no matter what gear you put, there will be no motion of the car. Only when the clutch gets engaged is motion generated. Okay. With that, I stop here for today, and I thank you for your attention.